Good morning. I'm Dr. Andrew LaBarbera, Chief Scientific Officer of the American Society for Reproductive Medicine. Welcome to the 19th presentation of the ASRM Grand Rounds webinar series. These webinars are designed to address topics in the ABOG Guide to Learning in Reproductive Endocrinology and Infertility. Today's presentation is by Dr. Richard T. Scott. Dr. Scott is the Clinical and Scientific Director of the Reproductive Medicine Associates of New Jersey and Professor of Obstetrics, Gynecology, and Reproductive Sciences at the Robert Wood Johnson Medical School. The title of his talk today is Assisted Reproductive Technology, What Does the Future Hold? I now will turn things over to Dr. Jeffrey Hayes, who will review the details of today's presentation.
Thank you, Dr. LaBarbera, and hello to everyone. I'm Jeffrey Hayes, the ASRM Education Specialist. Before beginning the webinar, I'd like to go over a few housekeeping items. First, after the webinar is done, please do not forget to return to ASRM eLearn to take the post-test and complete the survey for your continuing education credits. You must complete the post-test questions successfully and complete the survey to receive credit and be able to print off your certificate. Also, if you wish to ask a question to the speaker about the presentation, when you return to ASRM eLearn, click on the page link labeled Questions, and an email address will be provided. The question period will be only open for 3 weeks after this presentation is posted. After the time period for questions is expired, the questions page will be a frequently asked questions page pertaining to this presentation and topic. Our speaker today is Dr. Richard Scott. We are very excited for his talk, so I will now turn things over to our speaker.
Well, thank you very much, Dr. Hayes, and thank you very much for the opportunity to be here. We're going to talk some this morning about the future of ART, and the goals are really that everyone just sort of gain a better understanding of the areas that are actively being investigated in the assisted reproductive technology community, list some of those applications and what they might be used for, and then talk about new technologies which are coming into the arena which may also help patients help with their family building that are really outside the traditional realm of infertility. For disclosures, I am a member of a not-for-profit board, but I receive no personal benefit or payment of any kind from that foundation, and I have no other disclosures. So reproductive endocrine has been a really interesting field in the sense that when it really began back in the 1930s and 40s, we were really physiologists, and much of the study was about ovulation and things that maintain pregnancy. By the 50s and 60s, we became significant endocrinologists. By the 70s and 80s, we became surgeons, and if we could not bypass anatomic defects, we became cell biologists to do IVF. By the 90s and the 2000s, we really had progressed to molecular biology, and now beyond that to truly molecular genetics and molecular diagnostics. The future, who knows what that will hold, and that's a little bit what this talk will be about. Will we be altering the intrinsic potential of gametes, or simply refining our technologies and focusing on the best ways we can to allow each gamete, each embryo, to attain its own maximum potential? So exciting times and really a very rapidly changing field. In the future, there should be more high-quality RCTs. There should be more multi-center trials. We should have better statistical power and better statistical analyses for what we do. Right now, though, I'm talking about the future, so I get to cheat a little bit. I'm going to talk a little bit about the basic science, but it's still evolving. Translational studies are being done, but many others remain to be done. And of course, it's the future, so these large clinical experiences for things that are new are not yet available. But it's still really fun to think about, and I think these things will be here before we know it. Follicular simulation is an area that I'm not going to spend a lot of time on this morning. It is something that most reproductive endocrinologists have really excellent skill sets with. But in the end, we'd love to reduce the burden of care. I believe the data, at least as that which we have today, says that we're going to continue to stimulate patients for a long time. The average oocyte is not reproductively competent, either as far as we know in natural cycles or stimulated cycles or ART. And so we're still going to need a large number of oocytes to try to find that embryo that's reproductively competent, and the data today certainly support that. Oral FSH agents are coming. They may not be quite ready yet, but they're likely to get here at some point, and that should make it easier for our patients. Luteal support is going to become simpler, and patients may require less monitoring or even at-home monitoring for some of the things that will be part of the future. Nevertheless, the principles will remain the same, and many of the limits will remain the same. Where the future will bring us greater transitions is probably I'm going to speculate to say the near abandonment of fresh transfer, if not complete. We're going to vitrify, or whatever your cryopreservation technique of choice might be, during those retrieval cycles, and then replace those embryos back in hormone replacement and natural cycles. The reality is this analysis, meta-analysis, really demonstrates that you get better outcomes from ART cycles when the embryos have been cryopreserved and transferred in the fresh cycle. We're not really making the embryos better, and so it's likely that this reflects the importance of the endometrium and the milieu in which it develops. Now, just because you have a meta-analysis done doesn't mean that you have a definitive answer. We need larger studies and more diverse populations. But the progressive opinions of many clinicians in the evolution of this data is such that cryopreservation will become even more important, and avoidance of hyperstimulated endometrium will probably be the standard. At the same time, we're still thinking about things at the patient level. I'll point out that the microbiome is likely to find a big place in the future. This has just been a fascinating area of research that has exploded. While it has received more attention from our maternal fetal and obstetrical colleagues because of the impact it has on intraamniotic infection and preterm birth, the reality is that there's already quite a bit of literature to suggest that impairments in fertilization, altered tubal transport with altered embryo migration, potential chronic inflammation and altered implantation, as well as back to the very beginning, altered folliculogenesis and spermatogenesis are associated with changes in the microbiome. The microbiome really reflects the overall microbial content present in the body, and while there are microbes present in all those tissues, it's been found in follicles, follicular fluid, not during retrieval, but during open surgeries or laparoscopic surgeries when there should be little risk for contamination. And the endometrium, as we know, is certainly not sterile. And so alterations in that biome or potential systemic inflammation from the GI or other microbiomes in the body are likely to be important in the future. I'll put one pitch in for saying that looking at this is going to be more complicated than just doing cultures. And it's even going to be more complicated than simply looking at the genetics of bacteria to find out what's present, because the three-dimensional biofilms certainly alter the way cells interact and can potentially alter implantation. And biofilms from microbes are certainly present in endometrium throughout the cycle, including in the perinidatory interval. For those of you who may not have had much time to look at the microbiome literature, as I mentioned, it's really just now becoming visible in our field. It takes the advantage of the fact that the 16S ribosome in bacteria is very different than any of the genes, any of the genetic information which is present in the human system. So we don't have 16S ribosomes, we have 18S ribosomes, and so that are equivalently placed. And so if you look for a small amount of bacteria, even in the presence of overwhelming amount of human material, across the board, you'll be able to identify it because these genetic codes are unique. And then within those areas, there are a number of areas that are called the hypervariable regions, and these, in essence, create a genetic fingerprint for each species of bacteria. And there are over 5,000 distinct fingerprints for virtually every component of the microbiome. And so it's possible to sequence that area of the genome, particularly B4 and B6, and obtain valid fingerprints that allow you to know qualitatively which bacteria are present and quantitatively which ones may be contributing most to the microbiome in that individual patient or individual tissue. What's really amazing about all of that, again, is that it can be done. It's very reproducible. And as sequencing has gotten more practical, there are standardized protocols on a number of platforms, and there are standardized bioinformatics to help provide this level of sorting. And this just provides such better information that can come from culture. In fact, culture really only allows us to identify 6 to 10% of the microbes that are present in any given specimen at any given time, depending on where you're taking it from. And so we're really gaining insights. And in every tissue, in virtually every aspect of medicine and other parts of the body, this has been very, very important. And since 9% of the human microbiome is in the urogenital tract, it is likely to find some significance with us as well. Shifting gears a little bit, we're going to talk about the embryology laboratory. And I want to say that some of the most important things that are going to come are almost mundane. And so as we get new EMR and as we gain more connectivity in the laboratories, we're going to be able to use our electronic surveillance to also provide us really big jumps in quality assurance and quality control data. This graph is just an example from our own laboratory where we track blastulation rates across the laboratory as a whole. It is also possible to track blastulation rates of individuals who provided some component of care to that embryo, different incubators, different micromanipulation systems. You can isolate any component of the system. And by having these data automatically entered and automatically tracked, it's possible to have the software, the computer, actually tell you when you may be starting to see drift. And most everyone who's had the experience of having a significant diminution in their pregnancy rates in their program, everyone runs around and looks, and of course by the time you see the absence of pregnancies, you're a week or two past the time that the insults occurred in the laboratory, and frequently within a few days rates are back up and you really never know what went wrong at the time. The reason for that is because IVF laboratory data vary greatly. And so it's hard to measure variation in an endpoint, a clinical endpoint like blastulation or fertilization rates, or even subsequent implantation rates. It's hard to measure variation in something that has high intrinsic variability. There are Monte Carlo simulations. There's a number of statistical things you can do, a number of plots and graphs that you can employ, and then a number of rules like Westgard rules that can then subsequently be employed that empower the lab directors and the senior embryologists and clinicians to identify problems before they start impairing things, to allow us to determine optimal conditions to determine optimal outcomes. And these automated systems are really going to change the way we do QA and QC in the lab, and I think are going to be very important. At the same time, simple, mundane things like culture media and the basics of culture systems are not resolved. There's so many questions that remain to be answered. Oxygen tension, once 20%, now almost routinely applied at 5%, it still remains an unresolved question. In fact, there's really nowhere in the human reproductive tract where 5% is the oxygen tension of, which is attained in which our embryos grow up in vivo. And so some areas is 2%, others 7%. Obviously, it can vary. And so there are many good studies going on at many good centers, and we're going to further refine our knowledge of oxygen tension. Similarly, culture media. Amino acids, typically grouped by those as essential and non-essential, based on work done by Eagle on what makes fibroblasts grow well back in the 1960s, has really never been looked at systematically on a one-by-one basis or group-by-group basis to determine what mixes might be best for culturing our embryos under these conditions, and is, again, an area of active investigation in many labs. Growth factors, other macromolecules. There's a lot left to be done in this area, and it's likely that even though our culture media today are really outstanding and dramatically better than those of 10, 20, 30 years ago, the reality is there's likely room for improvement. Other adjuncts in the lab really remain to be investigated. It turns out the reproductive axis in human beings, the fallopian tube, keeps the embryo in an almost constant state of motion, and there are data now from some prospective studies and several large retrospective studies showing enhanced outcomes when embryos are moved periodically. All the time we've spent trying to hold everything perfectly still, it may in fact be better for embryos to have some motion during their culture, and there are a number of devices and a number of different platforms and different ways of providing that motion, though we still require further large reproductive outcome-based studies, preferably paired so patients could be compared to themselves and those studies are ongoing at the current time. Automation in labs is going to be another big part of the future. You know, if you go back 30 years ago, virtually every lab made their own media. They attained their own protein sources by going to labor and delivery and getting leftover fetal cord serum that was saved for them by the obstetricians and donated by generous patients who were delivering their babies. Now we have commercially available media and highly purified and highly screened, highly standardized, endotoxin-free protein sources. And that's reduced the workload in the laboratory and provided greater standardization. There are many other things that are going to get standardized in the laboratory. There are already automated vitrification systems available from more than one vendor. There are a number of people who are testing actually automated ICSI systems that can pick up the oocyte, isolate, immobilize, and pick up the sperm and potentially inject them to try to increase the precision with which that's done and the reproducibility with which that's done. None of those are really quite ready for prime time yet, but the reality is those days are coming. And there may be even simple things, making dishes, controlling drop size. If we're going to start looking in the drops to see what's coming out of the embryo to help us enhance our understanding of which embryos are competent, we're going to talk about that in a moment, then the reality is that we're going to need to make those drop size very consistent. And we're going to need to make the reproducibility between different laboratories or even different individuals within the laboratory a very, very reproducible phenomenon, which is very difficult to do now. Additionally, speed to avoid evaporation, because these droplets are likely to be slightly smaller than those used in most labs in the future, probably around 20 microliters, perhaps as little as 10 or 5, will mean that some standardization in devices will likely help us do that, and it will also just be faster and, again, reduce the number of embryologists we need or their workload. Will we be able to isolate embryos once they're fully cultured? Will we be able to grade? Will they load catheters for us? These are all things that people in different companies and in different laboratories are being pursued, but they're not quite ready for prime time yet. And I think we're still quite a ways off to the point where we're going to be ready to not have our embryologists looking carefully at our embryos for us and help making some of the timing and quality decisions of the future. I'm going to move on and talk a little bit about genomics, a little bit about where we are today, but mostly about where we hope to go in the future. Many of you have seen this graph or one like it. This is from Jason Fanasiak and was published in FNS a couple of years ago. And it shows the fact that embryos have a relatively low but still meaningful aneuploidy rate, even when women are in their late 20s, which is sort of the nadir of nature's curve, and that it increases steadily and reaches the high 80s to even 90% range by the time women are in their mid-40s. Across the board, this represents a significant impairment to reproduction, whether you're trying at home, whether you're able to conceive and have losses, whether or not you're infertile, even if it's tubal disease or male factor. The reality is that embryonic aneuploidy is a major cause of reproductive failure and a major component of reproductive senescence. PGS, as it's become known, where we're doing comprehensive chromosomal screening, seems like the latest and greatest new thing, perhaps over the last half decade. And the reality is that it's grown up over many decades of work by many distinguished investigators. In fact, John Rock, who did the original attempts at human IVF, although not for clinical purposes, wrote in an anonymous, but they believe it was written by him, contribution in the New England Journal that in the future we would be making and assessing embryos according to specifications. I think that many have followed in his footsteps since then to provide us with some of the foundational material. Bob Edwards were able to sex rabbit blasts back in the 60s, and Leander Wilton began doing biopsies, and Audrey Muggleson-Harris began the blast biopsies all the way back in the late 1980s. Yuri Valinsky did polar body biopsy. Alan Handyside had the first clinical case. Santiago Monet has made many, many contributions, and as has Treff and I think more recently Foreman. The reality is there have been many great investigators which have provided the foundation for this. This is not a new literature. People have seen the logic of this for a long time, both for aneuploidy screening and single gene defect screening, and it's a big part of the future. There are many studies. I'm not going to bore you with the past tense studies, except for one or two. And so we can really focus on where this may be headed outbound. So we've demonstrated that biopsies can be done safely, not so much on day 3, and the world is moving towards blastocyst biopsy. Those programs are there. But there it can be done safely without diminishing implantation rates at the current time. Additionally, we've shown that it alters selection criteria, and that the reality is in class 1 data shows you get better outcomes in this particular study and relatively good prognosis patients, but you get better outcomes when you perform CCS than when you don't in terms of clinical pregnancy rates, delivery rates, sustained implantation rates, all significantly higher. Additionally, for the first time, we're starting to see in the field performance of non-selection studies. And these are just extraordinarily important. One of the problems with dealing with human embryology is the finality of decisions which are made. We don't like an embryo because we don't like its morphology, we don't like its cleavage rate, we don't like anything about it. Those embryos are discarded. That's a very, very final endpoint. And the same is true with any genetics that we do. So if you're going to label this otherwise beautiful embryo as abnormal, you need a high degree of confidence that in fact it would not have made a baby. The only way to do that is a non-selection study where you literally will biopsy an embryo in this case, put it back, and see what happens to that embryo, which you can do through either single embryo transfer or DNA fingerprinting. Once you have the outcome, you go back and you analyze the specimen in a blinded fashion, and you say, hey, did this specimen predict that outcome? Did in fact, that biopsy that said aneuploidy correctly prognosticate that that embryo would not deliver? And with SNP arrays, it did it correctly about 96 or 7% of the time, but not perfectly. And with qPCR, that eventually came down to be in the 1 to 2% range of misdiagnoses. Now, those were not necessarily analytical errors, although they certainly may have been. It is also possible through things like mosaicism, some rare potential for self-correction, that embryos may in fact screen correctly in the sense of having that biopsy be aneuploid, even though that embryo has some reproductive potential. Still, the failure rate, the failure for an abnormal result to predict a lack of reproductive potential is very, very low and down in the 1 to 2% range. Marie Warner has recently done this with the new next-gen platforms. These studies must be done with every platform. They must be done with every significant technological change because the nature of the answers and the reproducibility and predictive value of these techniques must be validated again each time we change platforms. And when Marie did that, she was actually able in her initial cohort of just 41 embryos to have 0 misdiagnosis. In other words, every blinded embryo that was biopsied and transferred without waiting for results that was labeled as abnormal failed to make a baby. That study is just being completed. The numbers are now much larger, and the predictive values have remained exceedingly high. I think this slide just summarizes what applying these technologies can do for a program. These are just our local data. It doesn't really matter that they're from RMA, but if you go back to our 2005 timeframe, depending on the month, and each little data point here represents a month. The blue bars represent the number of cases we did that month, and the red line was the probability that a embryo that was transferred, these are just transferred embryos, the transferred embryos would go all the way to delivery of a healthy newborn infant. And back in 2005, it was in the 30s. It was in the high 20s sometimes. So 1 in 4, 1 in 3 embryos that we had done all that we could do for and that we deemed had a reasonably good chance of making a baby ultimately delivered. By looking at genetics and by also paying attention to synchrony, the things that Bruce Shapiro and Ernesto Bosch and others have taught us, that number is now approximately 2 out of 3. And so with that becomes great empowerment. With that becomes the ability to reduce the number of embryos we transfer while still simultaneously enhancing implantation and delivery rates. So this is the entire population. These are all transfers, all age groups, and across the board you can see that over the 10 years or so that this transition from no embryo diagnostics beyond simple morphology to use of embryo diagnostics in about 70% of our cycles we've seen dramatic improvements in outcomes, and we anticipate as new technologies come on board that the opportunity to select embryos is do nothing but improve, and we anticipate these numbers will get even better. Well, does that empower us to get to single embryo transfer? Some, the Europeans in particular, some of the Northern Europeans, would argue that we've always had the ability to do single embryo transfer. And they argue that you have to be very careful, and I think they correctly argue, to validate embryo diagnostics because while they do empower enhanced selection, and again, they also potentially empower discarding embryos that would have high reproductive potential. Still, if you do it right, you get higher delivery rates, lower miscarriage risk, you eliminate multizygotic multiple gestations, which is an enormous step forward in the field. And all of that in aggregate, you enhance outcomes and make pregnancy safer, get healthier babies. That's a win all around for the clinics, the docs, and embryologists specifically, and of course the patients most importantly. Why haven't we just always done this? And the reality is that people have tried. People have said if we're really careful and we really pick the very best good prognosis patients that have the best high quality embryos available, that we'll be able to get pregnancy rates that are just as good from singletons as we do for twins, because we're picking these ultimate elite cases with elite outcomes. And the answer is they did 6 randomized trials, and it's simply not the case. If you want to do multiple transfers over a longer period of time, you can in aggregate attain the same pregnancy rates, but not from a single transfer. And patients drop out from care after every transfer. People exhaust themselves emotionally, financially, access to care because they move. There are a million different reasons and a million different stories in different patients. But the reality is across the board, our obligation is to attain high fecundity while also being safe. And certainly just doing single embryo transfers based on morphology does not attain that goal. 6 randomized trials all showing the same thing. On the other hand, Eric Foreman did a study where he screened 1 embryo with chromosomal screening, ruling out aneuploidy the best he could, versus 2 unscreened embryos. Remembering that there'd never been a study showing that 1 embryo could equal 2 embryos in a randomized trial. And so in doing that, he found that there was no ultimate difference in the delivery rate, and that's an intent to treat delivery rate, not just a transfer. When you looked at the outcomes, 100% singletons in the single embryo transfer group versus 52 and 48, 52% singletons and 48 multiples in the control group. And of course, some might say, well, gosh, not so bad. There was actually 1 triplet in the control group as well from a monozygotic, but not so bad because patients love twins. And the answer is we shouldn't be so cavalier about that. If you looked at the birth weights, 3,400 grams average birth weight in the single group versus 2,750 in the 2-blast transfer group, and that included the singletons, not just the twins. And so I would challenge you to find an intervention in all of obstetrics, in all of maternal fetal medicine, whichever produces a 650-gram improvement in birth weight. I don't believe there's even one. I don't believe there's one that's done even half, or probably even remotely near that in terms of overall benefit to patients. Remember that ACOG has recently had a large program to encourage OBs to avoid elective delivery prior to 39 weeks. And their anticipated improvement in that in overall mean birth weight, 40 to 50 grams. And here we have the opportunity to improve birth weight by 650 grams, and it's just an enormously impactful number. If you look at low birth weight, 4% versus 32%. And I might mention that 4% is even a little lower, but it's pretty close to the general population risk, indicating that we can really normalize our patients' obstetrical outcomes. And very low birth weight, 0 versus 7.2%. So just enormous differences in outcomes, particularly in those latter 2 groups where the children are at risk for long-term sequelae. Do patients accept that, or do they just want to anyway? The reality is when you do CCS, these are our data from 2013 to 2015, you can get roughly 5 out of 6, maybe a little better now, to accept a single embryo transfer. And these are women who have failed 2, 3, 4 prior IVF cycles and they're still willing to accept single embryo transfer to avoid the high risk of multiples, which again approach 50%. In the unscreened population, we also push hard for single embryo transfer, but we're only able to attain 40% of our patients' consent. And so across the board, CCS not only provides higher delivery rates and lower loss risks, even on an intention-to-treat basis, but it helps encourage patients to make more fundamentally sound decisions that also produce healthier children. As technology marches forward, we're now to the point where we have other cool things. Mosaicism is coming down the pike. And you can see from this original paper from Mamas, it was published in FNS about 4 years ago, that they could see, and here it's chromosome 10. If you go from panel A to B to C to D, you can see that chromosome 10 progressively rises. And these were mixes of cells, and they created a standard curve from that. And from that, they felt that they could interpolate different graphs and identify patients who had mosaicism. Greco's paper from the New England Journal late last year really took this to the next level and said when you do that and you look for and identify those types of mosaicism, that if you transfer the embryos that are mosaic instead of considering them abnormal, they were able to get 6 deliveries in 18 embryos, 1 in 3 delivered. 2 had losses, 10 weren't pregnant, and overall in that group, because they were genetically screened, they average about 6 out of 10 delivering. So it's not as good to be at a third, but they're certainly seeing some pregnancies there. And it brings up the potential use of this information for counseling decisions to avoid them because they have a lower reproductive potential, but potentially not discard them as abnormal because they do have some very meaningful reproductive potential. The problem, of course, is that it's never that simple, particularly when something new comes along in terms of the molecular world. And so this is the actual data set that came right out of Greco's paper, the supplemental data, that showed how they created their standard curve for assessing aneuploidy. Over on the 0 on the far left, that's the variability of results when they used pure cell lines that had just all disomies. Now, they did it for 10, 100, and 1,000 cells. 100 and 1,000 cells are major league cheating. We never get 100 cells from a blastocyst. We'll never get 1,000 cells from a blastocyst. So really focus on the blue line. And when you do that, the author's own criteria in their paper was plus or minus 3 standard deviations. So let's create those standard deviations for them. The little gray lines for 1 and 3, for 2 and 3 copies, disomy on the left, trisomy on the right are there. But there are also confidence intervals for the mosaic mixes in the middle. But before we do those, look at that green box. The green box represents the confidence interval, 99%, 3 standard deviations, just as the author and other authors have utilized. And if anything in there can just be a pure disomy. Doesn't have to be mosaic. It could just be normal. And the 95% confidence interval for the trisomies comes right up to the edge. In fact, they overlap just the tiniest amount, but functionally they just abut with each other. Now put in the mosaics, which they did and are represented here by the purple lines. So clearly, mosaics fall in a range which is in between those of disomies and trisomies. Clearly, they overlap trisomic and disomic ranges, but it creates a problem in interpretation. They fall just where you'd expect them to fall. If you've got 50% disomy and 50% trisomy for a chromosome, you'd expect them to fall the range, their distribution results, to fall on either side, just like it does. But what happens when you get a level that comes back, average log-to ratio in this case, call it 0.13, how does one interpret that? Because remember, when you're evaluating a sample and you're using these curves to create your standard curve, you're working from the left. You're working from that axis. You look up your value, 0.13 here, and you say, well, gosh, that's in the disomy range. It's also in the roughly 1 in 6 or 7 cell per mosaicism range. It's in the 20% mosaic range. It's in the 40% mosaic range, and it's in the 50% mosaic range. The key here is that it's not about just speculating where you cross the curve, which is drawn by a regression line. The reality is that when you look at predicted values, it depends on the prevalence in the population of the abnormality And there's a lot of disomic, pure disomic embryos up in what I would think might be called the mosaic range. Not every embryo that's up there, however, is mosaic. It's greatly enriched with mosaics. There are a lot of mosaic embryos there, but it would be a significant mistake to label anything up in that range mosaic. You do not know with certainty that that embryo is mosaic. And so there's a lot of work that needs to be done here, and in the future we're going to do it. Why do we know it's important? Because when you transfer these embryos, we know they're greatly enriched with embryos that are not competent, because the implantation rates for these mosaics go down by about half. And the miscarriage risk actually goes up slightly in large clinical data sets, such as ours in the group from NYU. And so at the current time, we need to be cognizant of several facts. One of which is, remember, we're only screening a single biopsy. And the prevalence of mosaicism for any given chromosome is about 5, or for any given embryo is about 5%. Whereas we know if you look at the whole embryo, about 20% of them have mosaicism somewhere. Well, all we get is one embryo. We need that handful of cells that have a mosaic representation. And probably that's about 1 in 4 embryos that have mosaicism will be detectable that way. We need to make sure we don't assign diagnoses too comprehensively to these embryos. Their results are in the mosaic range. We know they are at increased risk for mosaicism. It is not sufficient at the current time to assign a diagnosis of mosaicism to those embryos. Certainly we can use these in selection and deselection. We need a lot more data. We need to have better ways to confirm these diagnoses, to separate the disomies and or trisomies from those that are mosaic. We need to know if they conceived of one of these embryos, what if it really was mosaic, what are the early risks in terms of loss and early development, what are the obstetrical and neonatal risks downstream. Certainly those studies are ongoing right now, and we're going to need them to make good decisions in the future. Same is true for duplications and deletions. This is a study Nathan Treff did using the Illumina platform and the VeriSeq data, which he has no interest in. Please don't fuss. And so the reality is that they're sensitive down to about 5 megabases in size. And in fact, we've been able to look at them on several different platforms, and they're all sensitive down to about 5 megabases in size. And of course, a lot of duplications and deletions, including ones with clinical phenotypes, ones with syndromes, are smaller than that, and we're not going to be able to detect them. Still, when you find them, this is, again, data from a prospective study done by Marie Werner, a non-selection study. There was almost a 50% reduction, almost 1/2 reduction in the implantation rates and progression to delivery of those embryos. So that's a very big difference. This phenomenon is real. What they're finding is real, and in fact, it does prognosticate altered outcomes. So these Denovo abnormalities are interesting. Again, we don't know if they're all real. We don't know if they're mosaic in nature and are non-contributing. We don't know with certainty whether or not they impact the inner cell mass. And we don't know the consequences of transferring many of these embryos. Even if they're meiotic, even if they affect the whole embryo, most of these dupe delts occur in areas that we've never seen in children, never seen even in miscarriage specimens. They're unique, and they occur throughout the genome. And so while some areas are a little more unstable than others, most of these are not syndromic, and we really don't know how to counsel patients. We can't tell them their child is not at increased risk, nor can we tell them that this embryo might not lead to a perfectly normal child. And so this creates a very difficult situation for clinical counseling. There's a great deal of data being accrued at many centers, and hopefully we'll have better data soon. That's going to be an important part of the future. A few gee whiz things to finish off my last few minutes here. We're going to learn a lot more about mitochondria, and I think it's going to go vastly beyond just counting mitochondria. I think there are data and have been data since the 2010 ASRM to say that that's unlikely to add clinically meaningful predictive value by simply counting them. High is certainly not as good as low in terms of these counts, but when you look at individual patients, their embryos tend to be all high or all relatively low, with only small differences seen, as we'll see in a minute. Across the board, I think the concept of evaluation of autophagy or mitophagy when it involves the mitochondria is a much higher yield area. And it's that dynamic which breaks, which allows accumulation of these high numbers of mitochondria, even though every measure of mitochondrial function actually shows less mitochondrial function in spite of these great numbers. And so There are great animal data to say that if you alter mitophagy in early embryos, that you, depending on the completeness of the block, you can get embryonic lethal, or you can get higher loss risk, or even losses with anomalies. And so our understanding of how to look at mitochondria, whether or not they're being recycled or shuttled out for autophagy correctly, is a big, big part of the future and is, again, an area of active investigation. I think whether or not our culture conditions impact that. Do they impact mitochondrial function? Do they impact mosaicism risk, or is that intrinsic to the embryo before we start? Is going to be a big, big thing. We see inter-lab differences. It may very well be that culture conditions do impact these risks, and that we can learn how to control those risks and improve outcomes. We also now have the ability to look at the epigenome in much more detail. The male work is tend to center so far on looking at the entire ejaculate. The group at Utah is doing some great work, and it's really very exciting. But across the board, we don't have the clinical predictive values or know how to translate that into active treatment. But across the board, the technologies are getting better, the precision of the data are getting higher, and this is a very active area of investigation. And we now have the ability to look at the epigenome in individual cells, at least over large segments of individual cells. In this case, it could be blastomeres or pieces of trophectoderm. And by looking at those, we're going to learn a lot about our embryos, which ones are competent, which ones are going down the appropriate developmental pathways, which culture systems produce the most appropriate development. And so this is going to be an exploding area, extremely exciting. Unfortunately, it's still very expensive to do these studies. They require very, very large data sets. One complete epigenome of a cell can require as much as 80 gigs of sequencing data, whereas 1 gig of sequencing can do aneuploidy screening on 46 embryos. So you can see that the great difference in the magnitude of information. But these studies are getting done, and it's really, really, really very exciting. Proteomics is changing as well. In the past, we've done immunoassays, and we've done some HPLC mass spec, and we've gotten sensitivities that are good, atomal range, even sometimes femtomal range, depending on the assay technique and what we're able to use. But mass spec is getting progressively better, and there's the new introduction of technologies. They've actually been around since the early 90s. These things called aptomers, which are actually small pieces of DNA that sometimes have synthetic purines and pyrimidines in them that are actually shaped to act like antibodies that can measure very, very low concentrations of analytes. And so you create an aptomer, like a receptor almost, that goes up and binds the analyte of interest, whatever you're trying to measure. And then there's a small DNA tail that extends off of this aptomer, which has a unique fingerprint, which means that you can detect as little as a single molecule. Because we detect single molecules when we do single gene PGD all the time. And so can we get in the octomole range? We really might be able to. The other thing is you can be extremely aggressive in terms of the breadth of your screening, because one drop of culture media can be screened for as many as 5,000 different proteins, and routinely for over 1,100. And so the reality is this can allow us for very high throughput screening, very high comparative screening, and across the board, we're excited that this may make a difference. Will it? Who knows? But the goals are there. And of course, we're not limited to just looking at the proteome in spin media. Antonio Capalvo and the group from Italy, they're showing that microRNAs, which are actively secreted by embryos, it's a normal event for microRNAs to be secreted. It doesn't require loss of cells. And they may be, there are associations with different stages of development, and they're pursuing whether or not they can predict outcome. Even if they don't predict outcome, when we're looking at proteomes or other lipids, other things that may be in media, we're going to have the ability to look exactly at stages of development based on that aspect of microRNA secretion, and it should help us do better studies. We're going to do better synchrony work. The work of Bruce Shapiro is going to tell us, again, that we need to be very careful in making sure that our embryos line up correctly with our endometrium. If we go to 100% cryo-wall, I think that's going to be very helpful. But we need to remember that this is important. We have to be very careful doing fresh transfers, especially in older patients who are at risk for asynchrony. This is particularly true if you do day 3 transfers because you don't know when that embryo blastulates. You really don't know if it blastulates in the window of receptivity or not, and it may be one reason that some programs do a little bit better with blastocysts. Looking at the endometrium to see if, in fact, it's not just a change in the hormone milieu creating problems, but something intrinsic to the endometrium itself, such as the ERA that's been proposed by the group in Valencia. But it could also be other things, proteome, secretome, metabolome, something we can measure in serum or cervical mucus. May also help us truly confirm that the window of receptivity is open and appropriately synchronous when we do our transfers. Many groups working on that. Recurrent loss is beyond the scope of this discussion, but I will remind everybody that recurrent loss patients, particularly recurrent biochemical loss, are not impacted by aneuploidy screening. And in fact, biochemical loss rates are not impacted by maternal age, which means we probably should have known all along it wasn't going to be impacted much by aneuploidy screening. And so what's causing those losses are areas of very active investigation, and of course, as we get more sensitive markers of the proteome and the metabolome, hopefully we'll gain further insights. Well, we find that there are true incompatibility issues. This has been talked about for decades between moms and dads. There's some work done originally by Susan Hibby in England, and then Juan Garcia Velasco in Spain, really showing that HLA-C type and CUR, which is a killer immunoglobulin receptor present in NK cells in endometrium. They're not related to the NK cells in the periphery, and you can't figure them out that way. The reality is that you can look at those, and Scott Moore from our group has recently shown that when you like to generally inhibit inhibitor or stimulate a stimulator, and in the past populations have evolved that way, but now that as a group we're interbred, we're getting mixes between the cells in the maternal interface that should be helping to dissolve parts of the maternal endometrium and create the lacunar lakes that will ultimately provide nutrition to the fetus, and the stimulus, which is presented from the embryo itself. And when you find mismatches across the board, you're seeing significantly increased loss risk. In fact, almost 2 to 1, even amongst euploid embryos and people with otherwise appropriate development. And so we may be screening our patients, particularly our third-party patients, where we have the opportunity to control that mismatch. PGD from expanded carrier screening. Expanded carrier screening identifies a relatively small percentage, certainly under 1%, of patients that are at risk for an autosomal recessive disorder or an unknown genetic disorder in their families. But if you screen enough people, that's going to contribute growth in the future. And so I suspect we'll care for more and more normal patients, especially now in terms of not being infertile, especially now because their outcomes are good, and including with single embryo transfers. I'll close by saying that fertility preservation is going to be a bigger and bigger deal. People are looking for markers to tell us who we should be doing fertility preservation on, who's at risk for diminished ovarian reserve by the time they're 30. Dr. Rebar showed years ago that 1 in 200 women go through menopause by age 32. Other groups have confirmed these numbers, and across the board, who knows when their ovarian reserve was declined, but it was likely in their 20s. We want to be able to identify those women in advance and tell them that they might be at risk, so that you might do directed fertility preservation. And then for the really big interventions, the what's new is old again. This is actually the third cytoplasmic transfer baby. It was a patient of mine riding a bike with her mom, quite a normal young lady. The first one is in graduate school. She's in the paper. She doesn't mind sharing. They're one of the big Ivy Leagues. These kids are great. But the reality is there were concerns about the safety and efficacy of this, but it's being picked up again. Variations are being done in England and other centers, and we'll see whether or not mitochondrial transfer or other appropriate interventions actually improve outcomes. For that, we will certainly need class 1 data. Will we be doing CRISPR or TALENs? Will we be editing and snipping genes? This is extraordinarily controversial. I doubt personally it'll take any meaningful role for those patients where we can screen embryos and identify which ones are healthy or low risk and which ones are not. But for disorders like mitochondrial disorders, where PGD doesn't really work, and where small variations, even very low level loads detected in PGD, which can frequently not be avoided, can still produce severely impaired offspring, really make the PGD not a good choice. Similarly, antenatal diagnostics don't work. Amnio, CDS don't really predict the mutation load in different tissues in children for many of these syndromes, such as MELAS. And the reality is that that might be the one place that you could do it safely. And there is work. The group from the Southern California at the Salk have shown that this can be done relatively well in mice. But there's still some off-target effects. And you never correct all of the underlying abnormalities. So there's some heteroplasmy remains, and the mutation is usually not completely cured. So I don't believe this is ready for prime time just yet. Plus, there are concerns for off-target effects. What happens if you edit the wrong place? Maybe the law of unintended consequences. You may solve one problem, but by creating a mutation elsewhere, you may be creating another. So this is important work, and it certainly is moving forward. Many are advocating clinical trials. They're being done overseas and outside the U.S., but as of yet, it may not. So in conclusion, there's a great deal of work to be done. Delivery rates are improving, which is great. Health and safety are improving for our patients. They're so much better than they were even 10 years ago. We all hope the care becomes less invasive. I really question whether it'll become simpler because I think diagnostics and precision and accuracy and genetic safety are going to be at a premium. And of course, those diagnostics will continue to receive ever-increasing focus. So with that, I would like to thank you very, very much for your attention and for the opportunity to be part of the ASRN Grand Rounds webinar series. And I'll be looking for your questions for the next few weeks, and I will certainly do my very best to answer them. Thank you.